Hi, I'm Mirjam from Bentum. I'm applying with my PhD thesis to the UPA Best Doctoral Thesis Award. This project is called Spike in Enhanced Phosphoproteomics Uncovers Synergistic Signaling Responses to MEK Inhibition in Colorectal Cancer. And the project was conducted at the Max Delbrück Center and the Charité in Berlin. In this project, I focused on a fundamental problem in phosphoproteomics, which is how do we reliably detect low abundance signaling peptides without sacrificing the discovery potential of global proteomics. So mass spectrometry lets us directly measure the phosphopeptides and read out the signaling network. But to study signaling meaningfully, we need both coverage of the relevant signaling nodes and high sensitivity, because many can be low abundance. And mass spec methods usually excel in one or the other. Global methods maximize coverage, and targeted methods like PRM maximize sensitivity. And to study signaling, we need both. And to this end, we developed Spike DIA, which allows you to have high sensitivity and high coverage in the same experiment. So it stands for Spike in Enhanced Detection in Data Independent Acquisition. And as the name says, it's based on DIA. In a standard DIA, low abundance peptides can fall below the noise and remain undetected. And in Spike DIA, I spike in heavy labeled synthetic versions of my target phosphopeptides before the measurements. And these heavy spike-ins act as beacons. Once the instrument detects the heavy peptides, I know exactly where to look for the corresponding low abundant endogenous peptide. And the result is a twofold increase in the number of target peptide identifications compared to your standard label-free DIA or DDA runs. And crucially, we still get a full global phosphoproteome from the same run. So it combines both targeted and discovery proteomics in a single workflow on standard instruments. To test Spike DIA, I applied it to MEK inhibitor resistance in colorectal cancer. When you block MEK in colorectal cancer cells, ERK signaling drops, but the cells adapt. And through feedback over multiple receptor tyrosine kinases, AKT gets synergistically activated. This was known. And then with Spike DIA, we could ask what other pathways and kinases might mediate MEK inhibitor resistance. Here we focused on KRAS mutants. And then using Spike DIA, we were able to identify that in KRAS mutant cells, MEK inhibition synergistically activates J and K through multiple growth factor receptors, and that this creates an additional resistance mechanism. To validate that this is functionally relevant, we combined MEK inhibition with J and K inhibition and measured cell growth. And the results was synergistic growth inhibition, exactly what the Spike DIA-based phosphoproteomics had predicted. So J and K activation upon MEK inhibition is both functionally important and therapeutically actionable. In summary, Spike DIA enables both network level coverage of the relevant signaling pathways. We used it as well as global proteomics. We used it to uncover a novel resistance mechanism through the J and K pathway. It's a modular method. It can be used with different panels, and therefore it's broadly applicable. The study is published in Nature Communications, and I invite you to take a look. And I believe that Spike DIA is a useful addition to the proteomics toolbox, and I'm looking forward to see how it will be applied.